My name is Isabel Blair, and for my seminar topic, I decided to study mtDNA use in DNA profiling. A little background on the topic. In more recent years, mitochondrial DNA has been observed with increasing emphasis in forensic science in terms of DNA profiling. While the great majority of DNA profiling techniques currently executed in forensic science utilize chromosomal DNA, abbreviated nuke-DNA, mitochondrial DNA is quickly earning a spot in establishing sources or profiles of a given specimen, and in some cases can even be advantageous over nuke DNA to use. However, using mitochondrial DNA has also brought about concerns of individual privacy and freedom among the greater public, something that must ultimately be decided. So, what is mtDNA? Well, in addition to chromosomal DNA found within the nucleus of the cell, a second human genome can be found in the mitochondria of the cell in the form of mitochondrial DNA. Now, while mitochondria have been traced to ancient bacteria that performed symbiosis with pre-embryonic cells, what exists now of the human mitochondria is a 16.5 kilobase pair circular genome. It has been determined through sequencing to contain of coding sequences for 37 different genes, and most importantly, a 1.1 kilobase pair non-coding region called the control region, or CR. What makes the control region so significant is its high variability among individuals comparable to the coding DNA. Because of this, forensic science in terms of DNA profiling and identification has focused foremostly on this very differentiated region of the mtDNA genome, with particular attention to the hypervariable region 1 and the hypervariable region 2, its two main polymorphisms. Application of the mitochondrial DNA genome in DNA profiling can be advantageous to chromosomal DNA profiling in two primary aspects. 1, copy number, and 2, the mode of inheritance. While nuke DNA is only present in 2 copies per cell, mtDNA is present in 500 to 2,000 copies per cell, which can be extremely significant in cases of small or degraded DNA samples. The second advantage of using mitochondrial DNA would be the maternal inheritance of mtDNA. Because it is maternally inherited, the sequence of such would be identical for all siblings and maternal relatives, greater than the immediate scope. One of the newer uses of mtDNA profiling is the prediction of the ethnic backgrounds of an individual. This is possible due to the maternal inheritance, high mutation rate, and lack of recombination observed in the control region of mitochondrial DNA. Because of the rapid evolution of the CR, specific mtDNA sequences are found in certain ethnic groups, but not in others. Forensic laboratories have placed together databases to compile and evaluate the accuracy of the mtDNA sequences, including databases used by the FBI and the Armed Forces DNA Identification Laboratory. An important concept in mitochondrial DNA profiling is exclusion versus inclusion. When an mtDNA sequence is used in forensic science, it is typically to include or exclude a particular individual to the sequence sample. By utilizing sequence comparison, an individual can be excluded indefinitely from a particular mtDNA sequence, but if an individual is included with the mtDNA sequence in question due to their specific sequence, it can be difficult to determine what this inclusion means. The problem is unlike exclusion. Inclusion is not a definite determination of source or not of source of the sample. This brings us to the question, what is a match? The significance of a match is solely dependent on the case in question. The mtDNA sequence can only be considered definite match if the potential sources are limited or restricted in some way. So, if in a closed population, identification through use of mitochondrial DNA sequencing would not be based on any sort of statistics, but on a process of elimination. because there are only a certain number of sources the sample could have originated from. Unfortunately, this is typically not the case, making mtDNA sequence matches not considered definitive identification scientifically or in the eyes of the law. The significance of the match must then be assessed to determine its importance by a set of guidelines. So, when sequencing DNA, there are two main focal methods. The most widely used method is known as the Sanger method, or chain termination method. The second method, which we will be focusing on, is the Anderson method. The first human mitochondrial sequence was described in its entirety in 1981 by Anderson, and so it is often regarded as the standard sequence when comparing mtDNA sequences. The Anderson sequence pertains to the light pyrimidine sequence, as compared to the complementary heavy purine sequence. Anderson's work have also led to the rules of nomenclature when referencing to mitochondrial DNA sequences. When determining the rarity of mtDNA sequence types, the counting method is typically used. The counting method refers to the number of times a sequence has been reported observed across various databases and has been judged acceptable within court cases in the United States. However, it cannot be used to determine the frequency of more rare sequence types, as the numbers cannot be estimated. Therefore, when utilizing the counting method, any figures determined are said to only be estimations and may vary from the actual frequencies. When considering mtDNA sequence matching, heritoplasmy and mutation sequences have to be considered, and in extreme cases, even homoplasy can occur, resulting from factors like genetic bottlenecks in the lineage or even among a single individual's tissues. As with all laboratory experiments, contamination is also a great source of error, especially because of the minute amounts used. There are a certain set of guidelines to validate mtDNA sequences in forensic casework, which I have listed off here. To read them off: if database sharing among laboratories is to be accomplished, each sequencing strategy should yield identical results for the same DNA sequencing templates. Maternal inheritance of mtDNA must be experimentally verified if it is to be used as a means of establishing identity in forensics. A number of tissue types from the same individual should be used for DNA extraction and sequencing to show that an individual's mtDNA sequence is identical from tissue to tissue, regardless of the somatic origin of the extracted mtDNA. Environmental insult studies, such as exposure of samples to the sunlight, soil, and so forth, should be performed to show that mtDNA sequence data is preserved and can be reliably typed. And lastly, with fluorescence-based automated DNA sequencers, a known positive control should be sequenced with each sequencing run. mtDNA sequencing has played a key role in reliably yielding DNA sequences in both extremely old DNA fossilized organisms and in unidentified human remains. Notably, these would include identification of Nicholas II, the last Russian Tsar, and Michael Blassie, the soldier buried within the tomb of the Vietnam unknown soldier. mtDNA sequence analysis was first introduced in casework in the United States in the state of Tennessee versus Paul William Ware in 1996, wherein a small sample of hair was left on the victim, and researchers were able to determine the origin, being Ware, by using the exclusion principle. However, with the introduction of mtDNA sequence analysis in mitochondrial DNA has come a number of ethical issues, the main reason being its ability to predict ethnicity or race of a particular individual. In 2008, Bush signed into effect the Genetic Information Discrimination Act, which hoped to keep the U.S. insurance companies and employers from discriminating on the basis of information collecting through genetic testing. This dilemma came from collection of bodily samples of individuals would leave in public considered abandoned DNA material. And this comes in the forms of saliva, hair shaft, fingernails, and so on. The public grew concerned about their privacy, especially as currently seizure of abandoned DNA material in public was legal by police, and is legal by police, who are allowed to collect DNA and store it in the premises of future crime detection purposes, regardless of whether an individual is suspected of a crime or not at the time of seizure. This too raises the question of how and when is it appropriate to create an mtDNA or mtDNA database without invading personal freedom and privacy by doing so, especially as databases are becoming increasingly connected with our current technological society. When would and when would it not be considered appropriate to conjoin current criminal databases and databases of non-criminals together? Should databases be available to public use in either situation, or should it be monitored exclusively by specific individuals like scientists or the government? These issues were of similar concern globally and were publicized by both the UK Human Genetics Commission and the National Council for Bioethics. Considering a sample could be obtained discarded DNA, to what extent would mitochondrial DNA sequencing be appropriate to determine ethnic background, particularly for discriminatory circumstances? Would it be appropriate to implement familial sequences or profile based on lineage? And is it still ethical to identify an individual through his or her mtDNA sequence? A relevant quote I found by the novelist Kay: "A warrant requirement will not make much difference to society that, under the sway of a naive and discredited theory of genetic determinism, is willing to lock people away on the basis of their genes. If the world is to make any advances in the field of mitochondrial DNA use in profiling, the opposing principles of security and freedom must come to some sort of balance." And here is my works cited. That is my presentation. Please let me know if you have any questions or comments about my topic, and thank you for watching.